But Kaylene, take it away.
Yeah, tag I'm it. Okay, so I get to follow on from Jack and tell you, I guess, a bit more about what stem cells are. So I think a lot of people will have heard about stem cells, obviously sitting here today. Jack's already mentioned them a couple of times, but they also pop up in the media quite a bit, and they're often discussed in a sense of, okay, stem cells are going to fix everything, and they could be used in so many different ways to cure so many different diseases. But I don't think that really tells us at all about what is a stem cell. It's a word that we've sort of all become so comfortable with, but maybe we don't really know what that means. So I wanted to take a couple of minutes to talk about that today. So I want to ask the questions: what are stem cells? Where do they come from? What is MS STEM? And how might stem cells lead to discoveries that will benefit people with MS? And so, can I get you to play the video for me, Jacob? I've been told if I go forward and then try and go back, I crash the whole system. So, okay. So if you look here, there's a cell in the middle of the screen that is down a microscope, and it's a single cell. And it is a stem cell. And what you're going to see is it's going to divide, and that'll result in 2 cells, and then they will divide, that will result in 4 cells. So we end up with more and more cells. And this is important because it actually comes down to, as researchers, what we define to be a stem cell. Stem cells are immature cells that will divide indefinitely. So they will just keep doing this and doing this and doing this, making more and more new cells. They do that because every time they divide, they keep themselves in their immature state. So they'll split in 2 and send another cell off, but they'll stay as they are, as that core cell that always has this ability to make new cells. But the other thing that's really important about them is that they don't just make any old other cells, they can make any other type of cell in the body. So the thing that makes a stem cell different from your hair cells, your skin cells, is that they have the ability to make every other cell type. So they could make blood cells, they could make brain cells, which ultimately is why originally they were only talked about in the context of embryos. So when we have a fertilised egg at the very, very beginning of conception of life, that is the ultimate stem cell because it clearly goes on to make every single one of us as people. So as we go through our lives, we started off as one cell, one stem cell, and we became a person. But even when we're like this girl here being, measured by her mother to see how tall she's grown, our muscles and our bones keep growing throughout life. And that is because we have tissue-based stem cells. So even in our muscles and our bones, we still have some stem cells that have the ability to divide and make new cells. But those ones are a bit different because they become restricted. They can only make, like the brain stem cells can only make brain cells. The blood stem cells can only make more blood cells. And so as we go through life, generally, everything becomes a bit more restricted. It's still amazing. There's still so much potential, but it does become a bit more restricted. So how is it that we are saying that we're going to get you to come into the clinic as part of our study? We're going to take a blood sample from you, and we're going to be able to use that to make all of the different cells. It's because it is now possible for researchers to obtain genuine stem cells, the ones that can make any cell type from the body, from adult human beings. And this was a discovery that ultimately resulted in 2 people winning the Nobel Prize in relatively recent history, because it's kind of amazing. And these stem cells are basically reprogrammed from already mature cells in the body. So we could say, please come into the clinic and give us either a skin cell sample or a blood sample, and we can take that into the lab and we can ultimately reprogram it. So we kind of push it almost backwards in time. It's like a little time machine. Put it into the lab, we give it some key genes that it needs, and what we end up with is a stem cell that we call an induced pluripotent stem cell, or an iPSC. And these are the cells that have come from adults, then they've come from your skin, they've come from your blood, and we've ultimately taken them backwards so they now have the potential, very much like the cells that start our lives at the very beginning, to make any of the cells that we want them to make in the body. And that's really important because when we're studying MS, we want to be able to study brain cells. And, you know, you can go into the clinic and give us a blood sample, but if I said, hey, anybody want to stop by the lab and give me a piece of your brain, I think all of you would be like, no, go away. And so in order to actually make this possible, we have set up MS STEM. Now, this is a bank of stem cells that we have generated from people with multiple sclerosis. And Dr. Ashish Mehta, who is around, he's at the back of the room, he's giving everyone a wave. He is the manager of MS STEM, so he is sometimes contacting people and saying, hey, please come and give a blood sample. And then he is taking that blood sample, doing the reprogramming and making these stem cells in the lab for us. We've been recruiting, and so ultimately we want 100 people with MS. We would like, ideally, this to be a mix of people with primary progressive MS and people with relapsing remitting MS, as well as people that are members of the families that Jack was talking about earlier, where the families that have this high incidence of MS. We also want some people that do not have MS, so sometimes that can be spouses, sometimes that can be siblings, because we also want stem cells from people that don't have MS, because otherwise when we do our research, we won't have anything to compare them to, so that we can't say, well, what's a bit different about these brain cells that was made versus these ones? We always need that comparison in order to do research. And so ultimately, this is what we'll be doing. We take the skin or the blood and we reprogram those cells, we make the stem cells, and then we take them through to make different brain cell types. This is just a picture of kind of what that looks like. We start off on day 0 with blood cells, and they get reprogrammed and they start to swell and get a bit bigger. And then we know the reprogramming's worked because they stick down onto the bottom of the dish and they start to spread out and have a different shape. And over time they grow into these colonies, which is them starting to divide and make more cells. And then, when we make them into brain cells, I think brain cells always look much prettier, this is what they look like. So this is an example of an immature oligodendrocyte on this side and a more mature oligodendrocyte in the green there. And then we can actually sort of study and say, what are they doing? How are they behaving? And the ones over the far side, there's some neurons that we've also made from some of these stem cells. And so the thing is that I do want to, well, I've got the moment I did want to sort of stress, because we ask people to come in and participate in these studies. And I'm extremely grateful, but I also think it's really important when we are talking about stem cells that people do understand that the stem cells can be frozen, then they can be thawed again and we can grow them, and we can grow them long term. And so when you do give consent to participate in these studies, your stem cells have the potential to be in a laboratory and even outlive you. And so this is something that people should think about when they give consent for this, because each of the use of the stem cells has to be done, they can only be used in a project where the person's gotten ethics approval, so they have to have actually applied and a committee has to have said, yes, that's responsible research. But, you know, this is something that I think a lot of people are like, oh, I'm giving a blood sample, you know, that's not a big deal. And, but I do want people to realise that, you know, we're keeping your cells for quite a while. Hopefully in the interim we will actually be able to use them to discover what's causing multiple sclerosis, and I think that that's going to be really going to be critical to us actually being able to cure MS. And so I'm going to skip that because I think... Yep, okay, now I'm going to jump back in at this point. So the value of it in terms of us being able to identify the cause of MS really does come down to what Jack was talking about, everybody's genome, is that the DNA, or I guess the genome, in the person with MS is exactly the same as the DNA that ends up in the stem cells, and then is the exact same DNA sequence that gets kept by the brain cells. So this is why we can actually then use these cells as a really good model for MS, because the genetic information that each of us carry is kept by these cells as in order for us to study them. And this may actually allow us to hopefully learn why immune cells attack the brain in people with MS in the first place. It's ultimately what we want to know. And so to get involved, again, there are flyers about signing up to participate and give blood samples for reprogramming. And I would like to thank various people who have contributed to this project, but obviously the MS community. The number of people that have already, said, yes, I'm happy to donate blood and to have that blood be made into stem cells for this project. We couldn't do that without you. So thank you very much.